Dr. Emma here to speak with us. She is a postdoctoral research associate at the Department of Psychiatry at the Washington University School of Medicine in St. Looking forward to having Emma visit us. Unfortunately, of course, we've had to delay the in-person visit, but we're very happy that she can still come and talk to us today. She received her PhD in psychology, behavioral, psychiatric, and statistical genetics, and now works in the areas of genetic architecture of substance disorders, and will be talking to us today about the overlap with schizophrenia. So Emma, when you're ready, feel free to share your screen with us and we can get started.
Thanks so much, Rachel. Wish I could be there in person, but okay.
You'll be among the first we have physically visit.
Yes.
Oh, thank you. Okay, is that working? Can everyone see my screen?
Yes. Yes.
Perfect. Okay. Yep. Great, and feel free to stop me at any time. I definitely don't think this will take a full hour, but thanks for having me. I'm excited to talk about some of the work I've been doing looking at the shared genetic overlap between alcohol use disorder and schizophrenia. I should disclose funding from NIAAA and AFSP, but no other disclosures. And I want to start with three general observations. So schizophrenia, of course, is a relatively rare disorder with a population prevalence of only about 1%. But within this group of people, the prevalence of alcohol use disorders can be as high as 40%. And this dual diagnosis is associated with a host of negative consequences, including longer hospital stays, higher rates of incarceration, lower treatment success, and medication compliance. So this is, of course, a serious problem. And in fact, up to 60% of early deaths in individuals with schizophrenia can be at least partially attributed to the use of alcohol and other substances. So what might produce this high level of comorbidity? And of course, the things I've listed here, these aren't mutually exclusive. So several of these could be true. The self-medication hypothesis is this idea that in order to relieve unpleasant schizophrenia symptoms or the side effects of certain antipsychotics, one might turn to alcohol or other substances. There could be shared environmental risk factors, so environmental things that contribute to an increased risk of developing schizophrenia, as well as alcohol use disorder. In addition, some of the impairments in cognitive processes that we often see with schizophrenia, these could increase the risk of developing an alcohol use disorder. And finally, there's some evidence that alcohol use disorder and schizophrenia might actually share some genetic pathways. And this is what I'm going to focus on today. We do see some evidence of genetic overlap between alcohol use disorder and schizophrenia. So both disorders, of course, are moderately to highly heritable. When we look at twin and family studies, we see a heritability for AUD of about 50%, and for schizophrenia, that's about 80%. In addition, when we look at recent genome-wide association studies of alcohol use disorder and problematic alcohol use, we see significant genetic correlations with schizophrenia. So the genetic correlation, or the RG that I'm highlighting here, is actually from Hong Zhou's most recent GWAS of problematic alcohol use. And the genetic correlation with problematic alcohol use and schizophrenia is 0.32, highly significant. There are also several polygenic risk score studies that show an association between liability for alcohol use disorder and schizophrenia and vice versa. However, when we look at the genetic correlation between schizophrenia and measures of what we might call more typical alcohol consumption, such as drinks per week, those genetic correlations tend to be a lot weaker. So the genetic correlation that I've highlighted down here at the bottom is from the G-SCAN study that came out last year by Liu et al. And when they looked at the genetic correlation between drinks per week and schizophrenia, that RG is quite low and non-significant. So there were really four main questions that I was interested in answering when we think about the genetic overlap of alcohol use disorder and schizophrenia, kind of driven by this prior evidence and some of these questions. The first, I was interested in actually examining some of the evidence for a causal relationship between alcohol use disorder and schizophrenia. To do this, I used something called a latent causal variable analysis. Second, I was interested in actually identifying variants with pleiotropic effects, so an effect on both alcohol use disorder and schizophrenia, as well as disorder-specific effects. Third, I was interested in partitioning the genetic correlation between these two disorders into relevant functional categories, as well as specific regions across the genome. And fourth, I was really interested in looking deeper at contrasting this genetic relationship between schizophrenia and disordered alcohol use with that for schizophrenia and a measure of more typical alcohol intake, like drinks per week. So before I move on, I just want to point out why we use something here called ASSET rather than a traditional meta-analysis when we conducted these cross-disorder associations. So ASSET is the software called association analysis based on subsets. And the idea here is that when you typically conduct a cross-disorder analysis between two different GWAS, you would use a traditional meta-analysis approach. And this can capture the effects of genetic variants that have the same direction of effect on both disorders. So, for example, a variant that increases risk for both alcohol use disorder and schizophrenia. But if you have a genetic variant that has a significant effect on both, but in opposite directions of effect, that will get washed out. So, for example, the SNP that I'm pointing out here is the most significant, what we're calling a divergent SNP in our analysis. So it has a significant risk effect on alcohol use disorder. It increases risk for AUD, quite significant. Conversely, it is highly protective for schizophrenia, so it has what we might call a protective effect. This is also highly significant. And in a traditional meta-analysis approach, this effect would get washed out. But thanks to ASSET can actually pool the effects of these variants that have opposite directions of effect on both disorders and use Fisher's approach to combine these into a single meta-analysis, z-score and p-value. So in this talk, first I will go through these four different aims that I discussed earlier, conclude with a summary and some next steps. So for this first aim, examining evidence for a causal relationship between alcohol use disorder and schizophrenia, I use something here called a latent causal variable approach, and this is a whole genome alternative to Mendelian randomization. So it only requires genome-wide summary statistics from previously published GWAS. And importantly, it can account for traits that are correlated, as we know that alcohol use disorder and schizophrenia are, and are highly polygenic. So it shows fewer false positives. And it defines causality in terms of this genetic causality proportion, what it calls the GCP. And this ranges between 0 and 1. So 0 means that there is no partial genetic causality between these two traits, to 1 meaning that one trait is fully causal for the second trait. And when we conduct an LCV on AUD and schizophrenia, the p-value for this null hypothesis that there is no partial genetic causality is 0.3, so we cannot reject this null hypothesis. Whereas the p-values for the null hypotheses that there's full genetic causality in one direction or the other are highly significant. So what we can conclude from this is just that we're not finding any evidence here of a causal relationship using this approach. I want to emphasize that we probably should look into this in more detail because this is not the only way to, of course, evaluate causality. But using this approach, we did not find any evidence of a causal relationship. So moving on into looking at
just
genetic.
Ask a question.
Mm-hmm. Yeah.
On that last slide. If you can't reject the null hypothesis, and the second bullet point would really be the alternative hypothesis, no?
Mm-hmm. Yeah.
So I see. So you reject the hypothesis that there's no relationship, and you reject the hypothesis that there's full causality.
Yes.
Okay.
Mhm.
Okay. So presumably there's some causal relationship. It's just not full causality.
I think, well, so for this null hypothesis of there's no causality, right? We can't reject that.
Right.
So from this analysis, there is no evidence of causality. Yes. Does that make sense?
Not completely. So the first bullet, you can't reject no causality.
Mm-hmm.
The second bullet, you can't reject. You do reject that there's either GCP of completely negative or completely positive causal relationship.
Yes, so either fully causal AUD to schizophrenia or schizophrenia to AUD. So this -1 and 1 is a little bit confusing. This is really just specifying whether trait 1 is fully causal for trait 2 or trait 2 being fully causal for trait 1.
I see.
Yeah.
Okay.
Sorry, that's a bit confusing.
It is. Thank you.
So with the second,
could everybody mute their line if they're not speaking?
So with the second aim, we're turning from causality to genetic pleiotropy. So here I was interested in identifying variants with pleiotropic effects as well as disorder-specific effects. In addition, we wanted to try and incorporate some gene expression data here. First, I'll take you through our European ancestry results. For our samples, we're using the PGC schizophrenia phase 3 GWAS, which has not yet been published, but hopefully will be soon. And this, we actually used a leave N out version, so this isn't the full sample. But our N cases here is about 50,000, controls a little bit over 70,000. For alcohol use disorder, we are combining two GWAS actually, this alcohol dependence GWAS and the PGC, and a GWAS of AUD from the Million Veteran Program. And our total cases here is about 43,000 and almost 200,000 controls. For drinks per week, we're using the G-Scan drinks per week GWAS by Liu et al. And the total N there is about 540,000. And one quick thing I want to note here, which I'll bring up in more detail later, we have similar numbers of cases between schizophrenia and alcohol use disorder, but note the very different numbers of genome-wide significant loci. So to identify pleiotropic variants, we use this approach called ASSET, and we use this to combine p-values from the AUD GWAS, schizophrenia GWAS. We input these into FUMA for annotation, and then we identified these subsets of SNPs, so what we're calling convergent SNPs that have the same direction of effect on both disorders, and divergent SNPs that have opposite directions of effect, as well as disorder-specific variants. And so we identified 24 loci with a risk effect on both AUD and schizophrenia, 24 with a protective effect on both disorders, 22 with protective on SCIZ and a risk effect on AUD, as well as 23 with a protective effect on AUD and a risk effect on schizophrenia, and then several variants with an effect only on schizophrenia, not AUD, and then only 3 with an effect on AUD but not schizophrenia. As an example of what ASSET is finding, here's our top convergent SNP. So this has a risk effect on both schizophrenia as well as AUD. And you can see here that this doesn't quite reach genome-wide significance in AUD, but it is highly significant in the combined meta-analysis. And again, here's that top divergent SNP that I showed you earlier, where we have a protective effect on schizophrenia and a risk effect on AUD. And then this is highly significant in our meta-analysis. Sorry, this plot is a little bit busy, but here I'm showing you the gene-based tests from MAGMA. We conducted this using the FUMA web-based platform. So on the x-axis here, you have chromosomes, and on the y-axis is the negative log 10 base p-value. And this is for our convergent subset of SNPs. So these are genes from the set of genetic variants that have the same directional effect on both disorders. And if we pull out the top 10 genes from that analysis and we conduct a phenome-wide association study, so essentially taking those top genes and looking at what traits they have been previously associated with in genome-wide association studies, we see that these have been previously implicated in schizophrenia and alcohol-related phenotypes, which is reassuring, as well as some risk-taking related phenotypes, such as number of sexual partners, risk-taking, whether they've ever been a smoker, neuroticism and ADHD, and educational attainment. So these are some interesting traits. When we look at the divergent subset of SNPs, again, these are the top genes. And if we pull out those top 10, we see these have also been previously implicated in schizophrenia, as well as alcohol-related phenotypes. In addition, we see educational attainment again, as well as some reproductive phenotypes and smoking phenotypes, of course, with the ChRNA genes. Now I'm presenting a gene property analysis. So this is also conducted through MAGMA, and this is essentially testing the positive relationship between tissue specificity and the genetic associations. So this is the convergent subset of genes, again, that have the same directional effect on both AUD and schizophrenia, and these seem to be enriched specifically in brain tissues. When we look at a pathway analysis, there were two significant pathways for the convergent subset of SNPs. So this reactome developmental biology pathway, of course, is involved in developmental processes, particularly regulation of pluripotent stem cells and activation of Hox genes. There's also this axonal guidance pathway, which is also an important aspect of development. We didn't identify any significant pathways for that divergent subset of SNPs or AUD-specific variants, but for the schizophrenia-specific SNPs, there is this MAP kinase signaling pathway that was highly significant.
Do you mind if I interrupt you with another question?
Yeah,
could you go back a couple of slides to show the nicotinic, that one? The ChRNA3 and 5 are obviously implicated in smoking. Those are nicotinic cholinergic receptors. And it's interesting that they're divergent for schizophrenia and AUD.
Mm-hmm.
Both are associated with elevated risk of smoking, but apparently these genes contribute opposite effects in the two disorders.
Yeah, which is interesting, right? Because you would assume that smoking would also be correlated with both schizophrenia and AUD.
And I think genetically it's correlated with both.
Mm-hmm.
I mean, I know it is. So these are just two genes that are not part of that genetic correlation.
Mm-hmm. Yeah, it is interesting. I think we could go into a lot more detail here.
A number of these have also been associated with alcohol use disorder. I mean, you mentioned alcohol consumption, but GCKR, for example,
mm-hmm.
and BANK1, which is on chromosome 4, if I recall correctly, are both associated with AUD.
Yeah, that's true.
Yeah. Thank you. I'll shut up.
No, please don't. This is helpful. So now moving to linking these cross-disorder genes with expression data. And so here we're utilizing a meta-analysis of EQTL summary data. And this is all work done by Dr. Manav Kapoor. And here Manav used summary database Mendelian randomization to test whether the effects of these pleiotropic genes are actually mediated by gene expression in the prefrontal cortex. And the idea here is to test whether the effect size of a SNP on the phenotype is mediated or dependent on gene expression. This can help us actually prioritize genes that may be more likely to be functionally relevant rather than just, say, assigning SNPs to genes based on physical position. And so some of these top genes, again, we conducted a quick little PheWAS, and these have also been implicated previously with schizophrenia, as well as immunological traits, cognitive traits, which was kind of interesting, and then some metabolic traits. And interestingly, we don't really see too much overlap between this analysis and what we're getting from our MAGMA data, which is somewhat interesting, but maybe not too surprising, given that these are fundamentally different methods of assigning SNPs to genes. We also tried to link our cross-disorder analysis to gene expression using a differential gene expression dataset. So we used RNA-seq data from postmortem brain tissue, the prefrontal cortex again, from individuals with schizophrenia versus controls, and individuals with alcohol use disorder compared to controls. And we did see that genes with convergent effects in our cross-disorder analysis were actually enriched in these differentially expressed genes when we look at schizophrenia versus controls. So there were 435 genes that were significant for the cross-disorder, as well as being differentially expressed. And our permutation p-value here was 0.008. Moving on to our African ancestry results.
But before you do, could you explain, how do you interpret this?
Yeah, I'm still thinking through that.
Oh, okay.
I mean, I think one thing I want to note here, I think in pretty much all of these analyses, unfortunately, we're a bit underpowered still for AUD, which is unfortunate. But, you know, I do think it's interesting that we're seeing these genes that are significantly pleiotropic for both AUD and schizophrenia also show differential expression in the brain tissue of individuals with schizophrenia compared to controls. I think that's nice convergent evidence, if you will.
So you have the GWAS summary statistics from Hangzhou's problematic alcohol use meta-analysis?
I do not have those yet. No.
Okay, so those should be available
Okay.
pretty soon, and that's a much larger sample.
Yeah.
But I think it includes. Let's see now. Does it include PGC? I don't know.
I think it does.
UK Biobank. Yeah, it does. It includes PGC and UK Biobank and MVP 1 and 2. So you may have to remove some cases to avoid overlap or handle that. But that may give you more power for that kind of an analysis.
Yeah, that would be nice. Yeah, because even though we have about the same number of cases here as schizophrenia and more controls, just using the MVP plus the PGC, still think we're underpowered for these. Seems to be driven mostly by schizophrenia things. But moving on to our African ancestry samples. So for schizophrenia here, we're using data from the genomic psychiatry cohort. This was Tim Bigdelli's paper that came out last year. And here we have about 6,000 cases and 4,000 controls. So unfortunately, it's quite small. For alcohol use disorder, again, we're combining the PGC and the MVP1, phase 1 AUD GWAS. So our total N cases here is about 20,000 and 42,000 controls. Due to this lower power issue in the African samples, we focused on just the overall set of pleiotropic cross-disorder variants. So I'm not separating these into convergent and divergent subsets. We did identify one genome-wide significant locus and one significant gene from the gene-based analysis. ADH4 came up. Since here it's kind of the opposite, we're definitely more well-powered for AUD. I think AUD is clearly driving some of these results. But interestingly, we did see some significant enrichment for early childhood brain development. Sorry, this is a bit blurry. And this is using the brain span data that's available on the FUMA platform. So unfortunately, these analyses, of course, are underpowered, seem to be driven by AUD right now. So next steps, of course, we'd like to get more samples, particularly for non-European schizophrenia samples. But I think the next step, we might think about conducting a trans-ancestral analysis. I think that would be interesting. Any questions there?
Hi, this is Teresa. Can you hear me?
Yeah, hi.
Hi, I just had a quick question about how you're measuring European and African ancestry, given that you may have some Africans who are straight out of Africa, you may have some African Americans, you may have Caribbean Americans, you may have Europeans of various immigrant status. How do you measure, and what exactly are you looking to see in ancestry?
Yeah, that's a great question. So I think all of these samples are likely African American samples. So I think all of these are based in the US. And all three of these GWAS use genetic ancestry principal components to define ancestry. So this is all based on genetic ancestry, not self-reported, if that makes sense.
Thank you.
That's a good question. So for the third aim, and I should be clear that unfortunately the rest of these analyses that I'm going to present are all in the European ancestry analysis because a lot of this secondary data that we use isn't available yet for non-European ancestries. So here I'm really interested in partitioning the genetic correlation and covariance into functional categories as well as specific genomic regions. So I'll actually start with this table on the right here. This is partitioning the genetic covariance between alcohol use disorder and schizophrenia into what we're calling functional and non-functional regions of the genome. And these annotations were defined using the GenoCanyon annotations. I don't know if any of you are familiar with that annotation framework, but they use biochemical annotation data as well as genomic conservation measures. And so basically their measure of what they're calling functional isn't whether a gene is coding for a protein, it's whether if there's a mutation in a variant that would actually have a phenotypic effect, an effect that we would see. And so both the functional and non-functional categories here are significant, but if you'll notice, the concentration in this non-functional category is nearly twice as much as in the functional, which I'm not really sure what to make of that yet. Still thinking through why that might be, but it is interesting. On the left here, we have the genetic covariance stratified by broad tissue type, and this is using something similar, the Geno Skyline annotations. I think these are both from Yale. And here we see that genetic covariance is significantly enriched in brain-related tissues. You might also notice that it looks like it's nearing significance in immune-related tissues. So that was close, but after correcting for sample overlap, that's not quite significant. Moving on, so here we're partitioning that genetic correlation or covariance into particular regions along the genome. So on the x-axis here, we have chromosomes, And this top panel, this is the local genetic correlation. So this is the correlation between alcohol use disorder and schizophrenia. The second panel is the local SNP heritability for alcohol use disorder. So that's why you see this on a different scale. And then the bottom panel is showing the local SNP heritability for schizophrenia. The first thing I want to point out, these two are on the same scale, but obviously the heritability that we're able to explain at this point in schizophrenia is much higher across the board than what we're seeing for alcohol use disorder. For alcohol use disorder, we do see this huge jump, this large proportion of heritability at the ADH1B locus on chromosome 4. Interestingly, we see a very significant negative correlation between AUD and schizophrenia at the regions immediately adjacent to ADH1B. We also saw this in the asset analysis, which I thought was somewhat interesting. So any questions with these analyses? Okay. For the fourth aim, I'm going to look at contrasting this genetic relationship between schizophrenia and alcohol use disorder with that for schizophrenia and what we're calling a more typical measure of alcohol intake, drinks per week. So again, as I mentioned earlier, there's been little prior evidence that schizophrenia and drinks per week is genetically correlated. But first, I wanted to actually recalculate that genetic correlation using these newer schizophrenia 3 sum stats that I had access to, as these were better powered than the phase 2 summary statistics that were used in this study. And then I wanted to actually test whether the genetic correlation between drinks per week and schizophrenia and the genetic correlation between alcohol use disorder and schizophrenia are significantly different. So the genetic correlation between drinks per week and the new schizophrenia summary statistics is still quite low, but this actually reaches significance now due to the increased power. The genetic correlation that we calculated between AUD and schizophrenia is 0.38, highly significant. And so unsurprisingly, we do see a statistically significant difference here between these two genetic correlations. And we can say that the genetic correlation between schizophrenia and AUD is significantly larger than what we're seeing between schizophrenia and drinks per week. I also stratified the genetic covariance between drinks per week and schizophrenia.
Can I ask a quick question just about that slide?
Yeah.
Is the implication there that drinks per week is not such a good metric for AUD? What is the implication of this? I guess.
Yeah, I think one takeaway, I guess the takeaway that I'm most interested in is that drinks per week, yes, is not a great measure for AUD, which is probably not going to come as a huge surprise to all of you. But I think there's been a push, of course, for larger sample sizes for GWAS, and that is important, but it's also important to make sure that we're actually measuring the phenotypes that we think we're measuring. And so something like drinks per week really isn't getting at the same genetic influences as AUD. They're just not the same trait. And we do see these different genetic relationships between traits like schizophrenia when we're looking at drinks per week versus AUD. Mm-hmm. And I think there's been evidence of that in the literature for a while now, but we just hadn't actually quantified that yet.
Thanks.
Mm-hmm. So I think this is just sort of driving that point home. When we look at the genetic covariance between drinks per week and schizophrenia, we don't really see any meaningful concentration in any particular tissue like we do for AUD and schizophrenia. So another way that I wanted to think about this was actually using polygenic risk scores. So I wanted to look at the association between polygenic scores for schizophrenia and a range of different alcohol outcomes in an independent sample. The idea here was to see if schizophrenia is more associated with particular aspects of alcohol consumption or disordered alcohol use than others. So I created polygenic scores for schizophrenia, and I should note that this is not using the newest schizophrenia phase 3 summary statistics. This is back to the older phase 2 ones. And this is a Bayesian method that uses an external LD reference panel to account for LD between SNPs. And our target sample here was the collaborative studies on the genetics of alcoholism, better known as COGA. And I should note that this is somewhat of an extreme family-based sample, so these are high-risk individuals. And the phenotypes that I looked at were some measures of consumption, numbers of beers or wine or liquor per week, maximum drinks in 24 hours, MAC drinks per week, the age someone first got drunk, whether or not they had a mother or father with AUD, their max AUD symptom count, and a diagnosis. And so I've sort of ranked these phenotypes here along the x-axis in what I thought might be the relative order of severity, moving from measures of more consumption per week to actual disordered alcohol use with an AUD diagnosis. On the y-axis here, we have the percent variance in the phenotype that's explained by the polygenic risk score of schizophrenia. And so you'll notice here that the schizophrenia polygenic score really only significantly predicts variance in these most severe phenotypes. I should note these are relatively small amounts of variance being explained anyway, but they are significant. I also decided to compare this with a polygenic risk score created from the PGC's latest cross-disorder effort. So this was a GWAS that combined schizophrenia, major depression, bipolar, anorexia, ADHD, autism spectrum disorder, OCD, and Tourette's. So this was kind of across the board. And here I'm only looking at AUD diagnosis. So on the x-axis here, now I have the different polygenic risk scores that I use, the schizophrenia risk score, cross-disorder risk score, and this cross-disorder risk score after controlling for a polygenic score for AUD itself. And what I found interesting was that this cross-disorder polygenic risk score actually outperforms the schizophrenia risk score even after controlling for a PRS of AUD. So perhaps not surprisingly, this does suggest that variants which are pleiotropic for the other disorders also show overlap with AUD. This isn't surprising given that this included major depression, which we all know is genetically correlated with alcohol use disorder. In addition, this cross disorder is much better powered than the schizophrenia GWAS that I was using to create this polygenic risk score. So this might change if we used an updated polygenic score for schizophrenia. But I did think these findings were somewhat interesting, and I would also be curious to see if this score for the cross disorder actually predicts variance in measures of alcohol consumption. So maybe it's not so specific as the schizophrenia risk score. Maybe schizophrenia is really the one that is specific to severe alcohol phenotypes. That's something I'd like to test in the future. So next steps, you all probably already know this, but alcohol use disorder is a highly polygenic trait, and we really do need larger sample sizes. And as I mentioned earlier, we need larger sample sizes of disordered alcohol use, not just measures of drinks per week, if this is what we're interested in looking at. So this is, again, another snippet from that row Hess plot showing the amount of SNP heritability that we're explaining for schizophrenia compared to a much lower level of heritability explained for AUD across the board. So what this really is suggesting is that alcohol use disorder shows a genetic architecture more akin to something like major depression than schizophrenia. It would also be nice to have more informative samples, of course. So, for example, we don't know how many of the schizophrenia cases from the PGC also have alcohol use disorder or another substance use disorder. And that would be a really nice thing to know. So we are working on that as we speak and hope to have more information on that in the future. Of course, I would still like to know more about the underlying biology. I think there's a lot of information in this data, but I think we're still only scratching the surface. And so it would be nice to be able to really dig down and know more about specific pathways and specific genes and what these convergent versus divergent pathways really mean. Another thing I'm interested in is examining which clusters of the psychiatric disorders from that cross-disorder polygenic risk score are associated most strongly with AUD and measures of disordered drinking. So for example, that PGC cross-disorder effort identified 3 different clusters of disorders. So one of those was mood disorders. And I think it would be really interesting to look at those polygenic risk scores and see which of those is most associated with particular aspects of problematic drinking. So in summary, at least from the preliminary analysis that we conducted, I didn't find much support for a causal relationship between these 2 disorders. But again, I think there are many different ways that you could look at that. Some people really swear by Mendelian randomization, and there are other causal inference methods that you could use. So I think we could really spend a lot more time doing a detailed analysis of the causal relationship there. When we looked at our cross-disorder analyses, we saw some differences in convergent divergent SNPs, significant pathways for the convergent subset in particular, as well as possible enrichment of those convergent genetic variants in differentially expressed genes when we look at postmortem brain tissue from individuals with schizophrenia versus controls. It's less obvious right now what the divergent SNPs represent to me, but I want to look into that in more detail. And then I'm happy to be able to present our African ancestry results, although they are currently underpowered at the moment, but I think that's a start. In terms of genetic covariance, we did identify a significant concentration of that covariance in brain tissues, which I think is nice, with some suggestion of potential enrichment in immune-related tissues, which is also interesting. Schizophrenia appears to share less overlap with consumption than with disordered drinking, which we somewhat replicated that in our polygenic scores analyses. Quick next steps for me. So a lot of this is based on my the aims I described in my F32. I was looking at the genetic overlap of AUD and schizophrenia. And that is almost over, but I also have funding from the AFSP. Here I'm more interested in looking at the genetic relationships between substance use, cognition, psychiatric disorders, and suicidal thoughts and behaviors, again using some of these polygenic score approaches. This all kind of leads up to my KO1, where I'm interested in taking some of these approaches and applying them to the genetic overlap between cannabis use disorder, and schizophrenia. Here, I think I want to focus a bit more on a potential causal relationship. This has been, of course, a big question in the cannabis use field for a while. So I think it would be really interesting to use genetic data to look at that, as well as pleiotropy. In addition, I'm kind of interested in trying to integrate a greater variety of multi-omics data, including some data from model organism studies to bolster some of these findings, and try to actually identify genes that might have causal potential for follow-up studies. Thanks to the PGC working group, the PGC schizophrenia working group, who has also been very helpful, as well as the Million Veteran Program and the GPC and GSCAN for letting us use their data, all of my collaborators and my co-mentors, and Elliot, and funding, of course. And thanks for having me. Any questions?
Question, please. Yeah.
Yeah.
In your reviews, did you come across any reports of the cocaine variable? In the 1980s and 90s, we had a very serious cocaine epidemic and seemed to be focused a lot in our schizophrenia population at the VA. Arthur Alterman was a member of the faculty at that time, and he had both VA funding and NIAAA funding for some of these studies that also heavily involved genetics. But he passed away, and I'm not sure whether his work ever got published. And I just wondered whether you had come across. We found one of the findings I remember very clearly because I was responsible for the treatment program, and there was a lot of evidence that the use of cocaine was aggravating schizophrenic symptoms as well as alcohol, and it's sort of a combination that made for severe clinical problems.
Yeah, in fact, that was Tom McClellan's paper that showed that people who used stimulants over years, including the amphetamines, achieved in the VA a diagnosis often of schizophrenia, and basically were often indistinguishable after about 6-year follow-on at the Coatesville VA. He looked at people who were long-term residential. So there was this interesting question about a pre-existing diathesis that would be aggravated by the stimulants. So it sort of raises the interesting question potentially of whether or not, Chuck took words out of my mouth, whether or not the overlap in vulnerability for stimulant use disorders and schizophrenia has been examined. This is from a non-geneticist, so I haven't looked at the literature in the way that you would have for schizophrenia.
No, that's interesting. I don't actually think I've come across a lot of that. I need to look into it. That sounds really interesting, though. You said that was Tom McClellan?
Yeah, Tom McClellan is the first author, and basically the irony was, and people had different hypotheses about why someone who has schizophrenia would use a stimulant, and they had hypothesis, well, if they're on their antipsychotic meds, maybe they're sort of feeling numb and shuffly from those that are blocking the dopamine system, and they would take a stimulant so they could feel something. But whatever the acute sort of feeling more alert or feeling some reward was, the longer term result was that basically it exacerbated symptoms and presented as essentially a disease de novo in people who were not initially presenting as schizophrenia. They were presenting as stimulant use disorder patients who then over years proceeded to a schizophrenia diagnosis. And I believe that might have been a New England Journal article. I could send you the reference. But anyway,
that's right. You're kind of merging a number of different studies, but the Tom McClellan study that you just described was in the New England Journal. The Arthur Alterman work was different from that, and it had separate funding and maybe some collaboration with other VA investigators, but I don't remember that clearly.
Yeah, I didn't know Arthur's data, but Chuck knows it. But I remember talking with Tom. I mean, the data had been collected for Tom's before it was there. It was when he collected the data and followed on with patients who were at the residential VA treatment center in Coatesville, Pennsylvania. So they were there, and it was sort of a good loop because you could follow them up over years. Anyway, I just was curious about it because of exactly the same things that Chuck was talking about. And I also just was curious in picking, obviously empirically there's this overlap, which is interesting between the two disorders. But since alcohol use disorder doesn't have nearly as much heritability as schizophrenia, would you be in a better position to choose one of the substance use disorders, like, for example, opioid use disorder, something as a comparison for specificity, but that does have an even higher heritability than perhaps alcohol? Help me with this, you guys who are geneticists. Isn't the heritability very low for cannabis and higher for stimulants and OUD and sort of intermediate for alcohol? You would know better than I.
I don't think the heritability for opiates is that high. I mean, I've seen high estimates previously, but I don't think the twin heritability is like schizophrenia, for example.
Yeah, no, I don't think anything is necessarily.
It's around 0.5 also, I believe.
The other issue there is that I don't think we have Well, I don't know what the largest sample size is right now for OUD,
OUD.
but I don't.
Hangzhou's is about
Okay.
120,000, including African Americans.
Okay.
That paper's in the press.
What are the data for those of you who know the genetics, and I don't, but for the interesting overlap between obviously smoking and schizophrenia has been looked at a lot. So how does that look in terms of paths, pleiotropy? What does
Yeah,
that?
that's a good question. That would actually be really interesting to look at. Because at least this is kind of a tangent, but when we look at alcohol consumption versus disordered alcohol use, we see these different associations with traits like educational attainment, cognition, but we don't really see those distinctions when we're looking at, ever smoking versus CPD, or nicotine dependence, as far as I know. So at least for smoking, it doesn't seem to have quite the genetic distinction between, lighter consumption versus something more like dependence.
Yeah, I mean, I know the
So,
rates of
yeah.
cigarette smoking are very high in patients who have schizophrenia.
Yeah.
And other interesting pieces that go along with that, with sometimes people talking about, maybe the nicotine consumption having a protective effect against Parkinson's onset, all sorts of colorful things that people have looked at that I don't have at my fingertips, but I just was curious about that.
Yeah, I haven't looked at that in particular, but that would be really interesting.
I had a question.
Mm-hmm.
Have you looked at socio-behavioral influences? I mean, I didn't see any demographics on your sample size. So what the ages of the people who participated in the study were. It's my understanding that schizophrenia shows up, in the early 20s. Is that correct?
Mm-hmm.
So
Yeah.
looking at the fact that that's the age that people start drinking socially, and
Mm-hmm.
what influence that might have on what you're seeing in terms of alcohol use.
Yeah, that's a really good point. Yeah, and I wonder if age as a variable, I don't know that that's available for all of our studies, which seems wild. It should be, but I would have to make sure. But that would definitely be an interesting thing to look at, especially if we're thinking of this in terms of the potential causal relationship.
I think age of onset of heavy drinking is highly correlated with the diagnosis of alcohol use disorder.
Mm-hmm. Yeah, unfortunately, I haven't really looked at any of those more socio-behavioral measures, but that would be really interesting.
What was your path, Rachel, in getting interested in schizophrenia? How did that emerge? Because it is a nice, highly heritable disorder, and there's a lot of data. How were you attracted to that?
That's a good question. Partly your first answer, but I started working on schizophrenia when I was in grad school, and I mean, I think a bit selfishly, there's been schizophrenia in my family before, so my mom and her parents, had lots of anecdotal questions and stories about why someone may or may not have gotten schizophrenia.
Yeah,
so I've just always found it, everyone just has these pet theories, and you really just want to know why, because it's such a...
Is it still considered a reliable fact about schizophrenia that in every culture where it's been studied, the prevalence is about 1%? And whether they give medications or not, witch doctors, whatever, whenever people have done anthropology studies in many different cultures, it always comes out about 1%. And I just wonder, that's dating from my training many years ago, and I wonder whether that's still true.
I thought there were a couple of very small isolated populations. Just remembering random papers I've reviewed, there are a couple very small, like isolated populations that have strangely high rates of like psychotic disorders and schizophrenia. But I think that is really rare, and it does seem to be hover around 1% in almost every other population. Yeah, that is interesting.
Yeah,
And
there are
used to be-
higher rates of psychotic disorders in some of the Amish populations, although that also so far appears to be due to common variation rather than rare variants.
And that's mostly bipolar, no?
Yeah.
It was bipolar in the family I was looking at, but there were other families who had psychotic disorders
Other than bipolar.
other than bipolar. Yes.
Yeah.
Yes, a schizoaffective disorder, that sort of thing.
Yeah.
Emma, I had a quick question for you in terms of the overlap of schizophrenia who had alcohol use disorder. So
Yeah.
would it be, if you had a sample where you had both, people with alcohol use disorder, people with alcohol use disorder and schizophrenia, people with just schizophrenia, and people with neither, what would you do? Would that give you more power for this sort of study? Or, I mean, I'm mostly thinking about the MVP.
Yeah.
When you have people with alcohol use disorder, and we could probably get the diagnosis for schizophrenia for a portion of them. Whether or not we could actually do that work, I don't know, but hypothetically.
There's an alpha project in MVP that's focused on schizophrenia.
Right. So yeah. So we could try and do a combined project where we. So what would you do with that sort of data?
Yeah, that would be a dream. That's an interesting question. Because I'm trying to think how you would want to draw your contrasts, right, between what's specific to schizophrenia, what's specific to the people who only ever, at least as far as we know, develop AUD, and then what is particular to these people who have both.
Right. Well, I'm trying to get at the idea of whether the genetic correlation that we're seeing is really because out of the people that have schizophrenia, you said, up to 40% have alcohol use disorder? Am I remembering that correctly from your...
Yeah, I think it's pretty high. Or it can be quite high.
It varies quite a bit across the samples, though.
If it's a sort of subset of individuals who are sort of driving these correlations, if we look at like people who never, who just have one or the other, maybe they might look different from the people that have both.
Yeah.
It may be that nature bundles them together quite often, which is interesting, but for statistical purposes and specificity, it would be great to have your schizophrenia population that doesn't have the other disorder that you're trying to understand. That would be great. Yeah.
Schizophrenia, alcohol use disorder in the context of schizophrenia is generally characterized by lower levels of consumption than in non-schizophrenics. So in other words, that may also help to explain to some degree the dissociation between measures of alcohol consumption and alcohol use disorder in relation to schizophrenia in terms of PRS, polygenic risk scores. But I mean, we see that in non-schizophrenic samples as well. So I don't think that's the whole explanation, but I think it may be, it may make that more extreme, the dissociation, that is.
That's a good point.
Are there any other questions or comments?
Hi, Emma. This was a great talk. I just had one suggestion. There will be coming online single cell transcriptomics from brain tissue for not only healthy controls, but several different CNS disorders. And you might then find much better relationships between the genetic risk and tissue expression if you're using a group of, let's say, neurons that have, let's say, the expression characteristics of excitatory projection neurons in the cortex versus just bulk tissue.
Yeah, that would be really nice. Is that through a particular study or?
Well, I think probably one of the people who's going to be publishing this first for schizophrenia, maybe Danny Weinberger at the Lieber Institute.
Okay.
But again, you still have the caveat that almost all of these postmortem brains were from people who perhaps took antipsychotics for a decade, and that's certainly going to be a large confound. But good luck with it.
Thanks.
All right, so it looks like we've reached the end of the hour. So thank you very much, Emma, for giving this talk virtually, and we do look forward to having you come visit in person and continuing these discussions.